Great. Thanks, Robbie. And how wonderful. So I need to stand here, right? To be able to. Yeah. So how wonderful to be here. This is the first opportunity I've had to visit the Institute, but we've had long contacts with the folks here, including Robbie. So really, really nice to be here. As Robbie says, I've been working on, I'm based at the Plant Breeding Institute in Sydney, which is outside Sydney, and I've been working on cereal rust now, me personally, for 38 years. 34 years of that at the Plant Breeding Institute. So you'd think after that long, you'd have this problem solved. Why do we still work on these things? Well, they are really, really smart pathogens. They know how to overcome resistance of plants. They change, so we're always trying to keep one step ahead of them. I will just advance the page down or enter. Thank you. So yes, these are pathogens that have been around for many, many years. They've been quite instrumental in shaping the wheat industry in Australia. In fact, so if you go back and look at European colonisation of Australia, they took wheat, the Europeans took wheat with them, and one of the first problems they faced was rust. So presumably they took rust with them from here, or we believe it may have survived on some native grasses that are susceptible. But nonetheless, they had massive problems with rust. This is before the rediscovery, this meeting was before the rediscovery of the laws of Mendel's laws of inheritance in 1891. This guy here, William Farrer, once graced our $2 note. We now have a $2 coin, so unfortunately we've lost the prominence of wheat on our currency, but Farrer was instrumental in addressing the problem of rust in wheat from a breeding perspective. And what he did was he actually developed wheats that matured earlier. So they weren't genetically resistant, but they basically escaped rust infection. And that was a huge step forward. It allowed them to push wheat production inland further where it was drier. And that combined with the earlier maturity of the varieties, it really made huge advances in reducing the frequency of rust epidemics. There's another guy here, Daniel McAlpine. He's a Scot. He's considered to be the initiator, I guess, of the field of plant pathology in Australia. We have an Australasian Plant Pathology Society, and the highest award they offer is the Daniel McAlpine Award. So yeah, some illustrious people here who made huge contributions to plant pathology and to rust resistance breeding. I don't know if you've read this book. E.C. Large is a British plant pathologist. Really nice book, just around the field of plant pathology, but that's a quote from that book. Basically, I suppose, drawing attention to the importance of these meetings. There were a series of these meetings. This is just one of them, this photograph. But the importance of these meetings in actually addressing the problem of rust in wheat. So as I said, Farrer was the first person really to try and address this problem through a breeding approach. We had McAlpine working with him, who was a pathologist. There's another chap there, Nathaniel Cobb, who devised the scale for scoring rust in the field that we use for phenotyping. Still use it these days, the Cobb scale. So that was a very, very important foundation and start for plant pathology and particularly rusts. And I'm still having trouble advancing these slides.
Yeah, if you click instead of using the side arrow.
Which is that, sorry, the touchpad or the?
Yeah, the touchpad.
Left one? Left one, got it. Okay. So, following on from Farrer's work, the University of Sydney began researching rust in wheat, and those studies were initiated by Professor Waterhouse in 1921. So last year we had our centenary of rust research. We've been conducting this research continuously at Sydney University ever since then. We had our centenary of rust research celebrations last year. There have been 4 leaders of the program. Some of you may know Bob McIntosh. He's quite prominent in the wheat genetics arena. Bob retired in 2000, and that's when I took over the program. Bob still comes in pretty much every day. So he's very passionate about wheat genetics, wheat improvement, and rust. My group at the moment, we have a great group of scientific staff and support professional staff, and you'll notice there postgrad students. We've got a very strong record in postgrad teaching. At the moment for us, this is a fairly small cohort of postgrad students. We normally have 15 upwards from all parts of the world, one of whom is tuned in today, Bosco, all the way from Uganda. Really, the numbers are down, I think, as a large part due to COVID, because a lot of our graduate students come from overseas. So we're in a rebuilding phase there. But I would say that our postgrad students have been very important contributors to our research output, and they've also been able to do some pretty amazing things. A lot of these graduates have gone back to their countries and are making huge contributions to agriculture there. So we're very proud of them. Okay, so rust pathogens, these are fungi that infect plants. You probably know that. Big problem in terms of biosecurity. They produce multitudes of spores. Those spores can be dispersed thousands of kilometres on wind. We've got examples of rust moving all the way from Central Africa across to Australia on high altitude winds. And once they get into crops, they really build up quickly, rapidly, just explosive epidemics. We do a little bit of work on rust on non-cereals. There's one there in the bottom right-hand corner, Myrtle Rust. This is a rust that's come into Australia in the last 15 years from South America. Our native Myrtaceae had no native rust pathogen, but we knew that they were vulnerable to this because there'd been some of this stuff grown in South America and infected. Well, we got it. It got into the country in 2012, and it's going to lead to the extinction in the wild of a number of our endangered Myrtaceae. So it's a nasty rust. Coming to the Australian Cereal Rust Control Program that I lead, it's a partnership between 6 organisations. They're listed across the bottom there of the screen. And that's basically been spun out or based on research that began at the University of Sydney all those years ago with Waterhouse. The kinds of stuff we're doing, in essence, we're looking at the pathogen and the host and then bringing that together, hopefully to help breeders develop rust-resistant varieties and also to manage those post-release. So an important part of that is to monitor the pathogens themselves. So we're doing race or pathotype surveys annually. We've been doing that continuously since 1921. So there's an amazing body of information there on how these pathogens have evolved over a very long period of time that's come out of that work. We have a genetics program. So this again is for all cereals, I should say. So we're working not just on wheat, we work on barley and on oat and on triticale and on rye. So that genetics program spans all those cereals. We're looking at discovering new rust resistance genes, characterising them, mapping them, developing markers, and increasingly now actually isolating the genes themselves to develop perfect markers and get a better understanding of how these genes work. And importantly, we have a link with all of the breeding entities in Australia. So those breeding programs for all of those crops have now been privatised. So we now have a very close interaction with all of the commercial companies breeding the wheat, barley, rye, triticale and oat. Most of that is now done under fee-for-service because these are commercial entities, but we test enormous numbers of lines in the field. I think this year we've got maybe 65,000 breeding lines in the field that we're screening for the breeders. They will come, we inoculate them with important pathotypes of rust. They come in, they screen that material and make selections. We also do germplasm enhancement, where we're taking the new genes that we find and then introgressing them into adapted germplasm and providing them to the breeding companies to use as donor sources of those resistances. And that's really, really important link for us. That links our research, the research we're doing. We're doing the teaching and training as well, which is also important. But that link with the breeding program is really, really important because that's how we get translation of our research into farmers' fields. So we have a strong research program, as I mentioned before, postgraduate students, really important contributors to that, postdocs and scientific or senior scientific staff. The 2 areas broadly we're looking at, rust pathogen biology, so trying to understand the pathogens better and how they are changing and overcoming resistance, and also host resistance itself. So I've already mentioned the idea of gene cloning, isolating the genes to work out how they function, and looking at new approaches like cis or transgenic approaches that might be applicable in future. So I'm going to give an example, a couple of examples of each of those, the rust pathogen biology, and the host genetics, just to give you some insight into the kinds of stuff we're doing. But before I do that, I just share with you some recent work we've done where we've been looking at fungicide efficacy in controlling rust. When I started this work, my career 38 years ago, farmers were just starting to use fungicides in broadacre crops in Australia. Prior to that, no one had used them. They just started using them, and the reason for that was that we'd had the appearance of stripe rust in Australia. So as I say, when I first started my career, it was pretty rare to find a farmer who would actually use a fungicide application. Now, it's pretty hard to find a farmer that does not. They're all using chemicals. They're getting chemicals produced in places like China off patent, very cheap, and they've got fairly broad spectrum against a range of fungal pathogens, so it's very unusual not to find a crop of wheat now in Australia or barley that's not sprayed with fungicide. So the question could be asked, given that, do we still need genetic resistance? Well, here's an example of why, one of the reasons why we do. So this is the barley leaf rust pathogen, Puccinia hordii. You would call it here brown rust of barley. We're looking at fungicide insensitivity here, or sensitivity to fungicides. With this, the fungicide we're using here is tebuconazole. It's a demethylation inhibitor fungicide that's used quite commonly in Australia. And with three different isolates of that pathogen. The first one was collected in 1969. That's pathotype 243. The one below that was collected in 1990, and the one below that was collected in 1997. And as you move across from left to right, number one is just a water treatment, and from 2 onwards you're getting increasing concentrations of tebuconazole. Okay, so you can see there, in that case, that these 3 isolates are being very, very well controlled by that chemical. 3 is even about a third of the low field rate that farmers are using, and it's giving very good control of those isolates. Here's an isolate that we found later, and another one, and you can see it's growing these isolates are growing quite well, even at the high rate, 1.3 times the high rate, and even higher, 12 times the high field rate of tebuconazole. So these isolates are insensitive. They've acquired insensitivity to the fungicide, and it's not controlling them anymore. And we know that that's actually not just tebuconazole. There are at least 3 other DMI fungicides that these things are insensitive to. So it's telling us that rusts have the ability to evolve insensitivity to rust, to fungicides, in the same way that they can evolve to overcome resistance. And I think that's a very strong reason why we need to continue to work on genetics and make sure that that is the foundation of our approaches to rust control. We found that that is related to we've actually found the genetic basis of that is related to a gene called CYP51, which is commonly associated with insensitivity to DMI fungicides in other fungi, including fungi that infect humans, believe it or not. But you can see there the 3 at the top, very low read coverage across that gene in one of the haplotypes. But then there are isolates below that that are all insensitive, and you can see that the read numbers jumps up. So we believe this is due to gene copy number, and we're verifying that at the moment. But that structural variant is very clear. We've sequenced, I think, about 40 or 50 isolates now, and we see that association in all of them. So we're quite confident that that's the molecular basis of that insensitivity. But again, as I say, this is clear evidence that these rusts can acquire insensitivity to chemicals, and I think it endorses the genetic approach to controlling them, reinforces that. The other thing I wanted to quickly mention was phenotyping. So this is really the basis of everything we do, and without good phenotyping, our genetic work really is not going to be very precise. And we're lucky that with rusts, they're really nice to phenotype. They're one of the easier diseases to phenotype. We do phenotyping in the greenhouse, phenotyping in the field. The guy there on the left from the field, you probably know, Brian Stephenson. I think he'd be known to some of you. We were scoring plots in Kenya that particular day, barley plots. Brian's barley material was stem rust. That phenotyping, yeah, it's really important. On the seedlings, we can tell a lot from the phenotype because the genes have different, what we call low infection types. All right, so here's an example of oat and crown rust on oat, and a series of known genes. Each one has a PC designation for Puccinia coronata and a number. just showing the typical low infection types associated with those genes. All right, so that's telling you a story. The genes will talk to you if you know how to interpret what they're saying. And so, for example, if you see a line that has an immune reaction like the one on the far left, you would say it doesn't carry, well, sorry, let me say that the other way around. If you see a response like you do on the PC91 line there, then you know that line cannot carry PC50 or PC68 because the low infection type is too high. There's necrosis and there's pustules forming. All right? To run you through an example of that, here are 3 oat varieties inoculated with one pathotype of oat crown rust. Here is the same 3 oat varieties inoculated with a different pathotype of oat crown rust. And here is the same 3 oat varieties with a third pathotype. And you can see that apart from the one for number 3, which is susceptible, we would consider to be susceptible to all 3 pathotypes, the other 2 are resistant, all right, to all 3 pathotypes. But the low infection type is different to each pathotype. And based on that, we can make some fairly specific hypotheses that we can be quite confident about without doing any genetic analysis, right? So this is just based on the phenotype. So the conclusion that we would make from this is that Baku, that's oat variety number 1, carries 3 resistance genes, all right? One that gives this what we call a NORT or immune reaction to pathotype number 1. Another gene that gives a flex C reaction, more chlorosis, to pathotype number 2. And then a third gene that's giving a necrotic reaction to pathotype number 3. All right, so there's no genetics here. This is just basically using what we call the gene for gene hypothesis and looking at the phenotype. And it's telling us that more likely than not, Baku carries 3 resistance genes. Betong, on the other hand, we would say carries 2 genes. So it's giving a NORT reaction to the first one, and then this necrotic reaction to the next 2. All right? And then finally, and probably the most important conclusion that we would make from those images, those phenotypes, is that Betong and Baku carry the same gene that protects them against pathotype number 3. Now, this is important because what it suggests is that a single mutation in pathotype 3 would render both varieties susceptible. And that's exactly what happened. About a year after I did this work and took this photo, I got a mutant from pathotype number 3 that rendered both varieties susceptible. And they were the last 2 oat varieties at the time that we had with any resistance to crown rust. So again, that was based just on phenotyping. No genetic populations or anything else. We were able to make the prediction, and it was verified with what the pathogen did. So anyway, that phenotyping is really important for us. It's the basis of everything we do, our genetics, our race analysis, and everything. And I'm very lucky to have worked in a laboratory that is really strong in this phenotyping, and I've learnt it. It's sort of corporate memory that I learned from my predecessors. It's been handed on through the generations. Okay, so now I'll go on to give you a couple of examples of the rust pathogen biology that we're working on, and then the host genetics. Okay, so rust pathogen biology, the 2 examples I'll give you, excuse me, wheat stripe rust and barley grass stripe rust, some recent changes we've had in those pathogens in Australia that have had big impacts in our wheat industry and in our barley industry, or potentially in our barley industry. So one of the things about Australia, it's a long way away. It's very isolated from other wheat growing parts of the world, but that's a good thing when you're looking at rust. So we have a closed system, and we can monitor these rust pathogens pretty much within a closed system and watch how they evolve over time and keep track of things very, very well. We started this surveillance back in 1921. Wheat rusts do not undergo sexual recombination, so all the variation we're seeing is pretty much coming from asexual processes, apart from occasional introductions of exotic isolates. So what we see periodically, we get an isolate turn up that's completely different to anything we have. It's come from somewhere else, it's come into the country, and then over a period of time it will just undergo mutation to acquire virulence for resistance genes in wheat varieties. And what we found with our resistance breeding is that we can do very effective preemptive breeding against those local mutants. We can sort of preempt what's going to happen next. But when it comes to these exotics, we've been far less successful. We don't know when they're going to come in. We don't know where they're going to come from. We don't know what their virulence is going to be, what their impact is going to be on not just current varieties, but also the material that's in breeding pipelines. And sometimes we get these incursions and some of the breeding companies lose a lot of germplasm from their pipelines. So to give you an example of that, as I said, wheat stripe rust. So we're monitoring these pathogens all over Australia. The samples are sent to us. We undertake the race analysis. We post that information on a map. It's updated weekly. Last year, that map had over 5,500 views. So we know that this information is valued by industry. And this is what we found over the last 100 years, that we've got a very strong trend towards increasing exotic incursions. This is just wheat rust, okay? So you can see there since I've just arbitrarily started this at 1915 and made it 20-year periods, but you can see that the frequency of these incursions is increasing with time. And in the current 20-year period, we're only 7 years in, and we've already had 3 incursions. So the trend is continuing. And we think this is largely due to people travelling more and people bringing rust in on contaminated clothing, unfortunately. But that's what appears to be happening. This is those 15 exotic incursions that we picked up of the 3 different wheat rusts. Last incursion of stem rust, 1969. Stripe rust, a very different story. So we had our first incursion of stripe rust in 1979. We then had a second incursion in 2002. And then we had 2, one in 2017 and one in 2018. And in each of those cases, these things have caused have really cost the industry a lot of money. So as I said before, loss of germplasm from breeding programs, loss of varieties. With this one in 2002, there were 3 years in a row where we had really big epidemics of this pathogen, this pathotype. And it was estimated that between $40 and $90 million of fungicide was put out specifically because of that incursion in each of those years. So they're costing significant amounts of money. The challenge that we've got now is that we've had 2 incursions of stripe rust, 2 really different pathotypes. And that is something we've never had to face before. We've had these incursions in the past. In fact, back in 1969, we had 2 incursions of stem rust, very quick, almost at the same time. But those 2 pathotypes were almost identical, which meant resistance, their impact on varieties was the same, and resistance breeding, the response to them was the same. We're in a very different situation now. We've got 2 different pathotypes of stripe rust, completely different in their virulence, completely different in their impact in our germplasm. And it really has caused problems, particularly last year, and is causing big problems now. So here's those 4 incursions of stripe rust. We've gone and done whole genome sequencing using various marker sets like SSRs and whole genome SNPs, and we've been able to put these into molecular or clonal lineages and determine where they came from, each of those 4 incursions. And this is the 2 we've got at the moment. S10 and S13, I'll call them. They're molecular or multi-locus groups. They're very common in Europe at the moment. And S13 is very common in South America at the moment. So we know that this S10 group came from Europe, and the S13 group either came from Europe or from South America. When these things appear, we can look at their spread, and every time one of these things appears, you think, well, can you eradicate it? Well, usually they spread so quickly it's not possible. So here's the 2018 incursion. It was first detected in August, and I think that's about 3 months later, we'd isolated that from locations more than 1,000 kilometres apart, including Tasmania. So these things move really, really quickly once they get in. And then the following year, 2019, 2,300 kilometres apart. So spreading very quickly. And this was in 2 years of drought, right? So these were really dry years, not favourable for rust, not favourable for wheat, not favourable for rust. And yet the rust still managed to establish and spread over a huge area very quickly. In 2020, this is the distribution of those 2 different pathotypes. The one on the left is this S10 or pathotype 239 that came in 2017. And the one on the right is the S13 or pathotype 198. And you can see that the 198 was far more common than the 239, despite the fact that it came in a year later. And And so what we found in 2020 was it was a return to good seasonal conditions. Farmers were getting great crops, stripe rust just exploded, and most of the problems were associated with this 198. So that was fine, we were able to explain that, but many varieties were resistant to 198 and they performed quite well in 2020. But then in 2021, for some reason, 239 built up in the pathogen population, and those varieties that were resistant or performed better in 2020 started looking pretty sick in 2021. And a lot of farmers were saying, what's going on? We said, this is because this other pathotype has increased in frequency. So you can see the dynamics of these two pathotypes is changing over time, and that's driving a varietal response, what we're seeing in farmers' paddocks, and decisions to make whether chemical intervention is required. And the same thing's happening this year. We've got stripe rust, an even bigger stripe rust year this year in Eastern Australia. Both of these pathotypes are being identified in the samples we're having sent in, but there's two more that we've now found, a mutant of the 198 that's causing a few problems, and another variant that we think may be affecting a variety that was resistant to both of those pathotypes. So I had one of the breeders contact me just when I arrived here in the UK a couple of weeks ago, saying they had big concerns about this variety. So we've already planted material in the greenhouse to look at those samples to try and work out what's going on. So that's the examples of wheat stripe rust. I'll give you an example now of the barley grass stripe rust pathogen. This is another interesting one, and one that I'm very concerned about. It's not causing problems at the moment, but I think we're in a situation where we may be going in the direction of increased problems with this. So what is barley grass stripe rust? Well, these rusts, some of these rusts have different forms or form of species that are adapted to different crops. Okay, so wheat stripe rust we call former species Tritici. It has high infectivity on wheat, very low infectivity on barley. And on this barley grass, this is Hordeum leporinum and Hordeum glaucum. These are weeds in Australia. It has very high infectivity and is present in Australia. There's a form that is specialised to barley, the stripe rust. It's here in the UK. We do not have that in Australia. Very high infectivity on barley, low on wheat. We don't know what it does on barley grass. We don't have it in Australia. We don't want it because we know that our barleys are very susceptible to this. We've tested them offshore, and we know there's huge vulnerability in our barleys to this pathogen. So it's another one of these exotic rust threats we don't want to come into our country. This guy here, the third one, we call pseudo-Hordii because it's a little bit like the barley stripe rust pathogen, but it's sort of got low infectivity on barley, a little bit more than, let's say, wheat stripe rust, but still low, very, very low on wheat, but high infectivity on this barley grass. We picked it up first in 1998. We believe it was an exotic incursion from somewhere. We've now, through genome sequencing, we know that it's also present in the US. So it may have come from there, we're not sure, but we know it exists elsewhere in the world. So it has not evolved locally. This is an article I wrote for one of our rural publications I wrote it in July 2021 based on our experiences of the past year, just telling people that there was stripe rust in some crops, the barley, not much, but don't worry, nothing's changed. We've been looking at this barley grass stripe rust pathogen for 23 years, haven't found anything, so don't worry too much, right? Well, I think it was, when was that? July, I think about 2 months later, I found a variant. So just after 23 years, nothing. I write an article and about 2 months later, I find a variant, which is why I say don't trust rust. So here are 7 Australian barley varieties infected with a pathotype of wheat stripe rust. You can see they're all highly resistant. This is with a different pathotype of wheat stripe rust. This is this S13 group. They're all still resistant, but you can see there's been a shift in the low infection type, right? So this thing has different virulence on barley, but it still can't really infect barley. This is the PGYR isolate, the original one from 1998, and you can see, again, they're all pretty much resistant, but that low infection type has crept up again, more vulnerability, right? And this is the new one. It's taken them all out as seedlings. So looking at those phenotypes again, the all-important phenotypes, I looked at this and I've done testing with it, and I've made the prediction that this thing has rendered 3 genes in barley ineffective, just based on phenotype. We're now doing some genetics to try and confirm that. But the phenotype is telling me that 3 genes have gone in our barleys. We're not seeing significant amounts of this in our barley crops, but there is more samples coming in on barley, low levels of infection, so no yield losses yet, but this is a significant change to increase virulence. And I'm very concerned about it because we don't know what this new one has to do now to start knocking over our barley varieties. But whatever it has to do is much less than what it had to do before in terms of acquiring virulence. And I tested some of the advanced breeding material, and there's shifts towards greater vulnerability, let's say, in quite a lot of that germplasm. So again, this shows you the link between what we're doing with the surveys. We identify something that's new, and then we need to work out what its impact is likely going to be in agriculture. And this is what we do. We take these isolates, yes, it's new, we then start testing germplasm to work out what the impact is going to be and to try and predict what's going to happen. So what I'm doing at the moment, we've crossed a lot of our barley varieties by a stripe rust susceptible, and we'll just run them out to F2s and test them with the new one and see how many genes are there. And if it's only one, we know we're in trouble. If there's lots of them, then we can be a little more confident that this thing's going to have to do a lot more work before it's really going to start creating problems in our barley industry. So what is that thing? It's actually also taken out a couple of genes in wheat. So here's a dendrogram based on whole genome SNPs of 10 isolates it's 8 isolates of wheat stripe rust, and then the 2 BGYRs. And you've got the 4 multi-locus groups there, all clustering together. BGYR is up here, and our new one's down here. So that's telling me this is not a mutant. It's not just a mutant, a spin-off out of BGYR. It's telling me it's probably an asexual hybrid between wheat stripe rust and the barley grass pathogen. And so that's a big jump, a big change in virulence. It's either got to be that or it's got to be another exotic incursion. I suspect that it's a hybrid. So we're now doing whole genome sequencing on this thing, and we're going to do chromosome scale assemblies to try and see if we can figure out what's actually going on in the pathogen to give rise to this big shift in virulence. Okay, so that's the rust pathogen stuff, and also how we sort of link that to controlling rust in agriculture. Now I'll talk a little bit about the rust genetics, and I'm going to focus here on our barley rust genetics, given that there's interest in barley rust, or barley genetics here. We do an enormous amount of work on wheat rust genetics. Robbie asked me yesterday, what would be the proportion of the time you spend on barley? Is probably about 15 or 20% compared to wheat. Wheat's the big one. But we've done some fairly nice stuff with the barley rust genetics. So I started working on this pathogen, Puccinia hordii, back in 1989, and I've had a series of students that have worked on this with me, and also some really talented postdocs and scientists as well that have made really, really important contributions. So I just acknowledge their contributions here. Some of the highlights from that research, well, I've been conducting the national pathotype analysis for this pathogen since 1989. We've picked up virulences, important virulences on these genes. So the leaf rust resistance genes in barley have the designation RPH, resistance Puccinia hordii, with a number, and each number indicates a separate locus. So I've picked up virulence for those genes. We did the first haplophase genome assembly for Puccinia hordii, so we're doing genome sequencing and looking at genomics of the rust pathogen. And as I showed you earlier on, we've demonstrated fungicide insensitivity in that pathogen. On the host side of things, we've discovered and designated 5 seedling resistance genes. So all these RPH genes we found, we've mapped and numbered. We started looking at adult plant resistance to this rust in barley and found 3 QTLs, which we Mendelized and have developed codominant markers for, and demonstrated additivity. So these we believe are probably durable resistance genes to leaf rust in barley, and we've now assisted the breeders with selecting them in their varieties. And we know that Australian barley varieties, quite a few of them, carry 1 or more of these genes now. We've also done a lot more work on the adult plant resistance. We've found at least 3 new sources that we know based on our mapping that must be different. And one of those is a recessive pleiotropic locus that we're very, very interested in and really doing a lot of work on at the moment. That's particularly interesting. But what I wanted to talk about more today was the actual isolation of these rust resistance genes. So we're now in a position where we can go in and pull them out of the genome and look at their structure. So my group has actually now cloned 4 of these genes in collaboration with other people, and we actually demonstrated synonymy of the gene RPH15 with the gene that had been previously designated RPH16. So I'm going to tell the story, I'll just make some general comments about the rust resistance genes that have been isolated from barley so far. So some of these have been isolated by other groups, some by my group. The interesting thing is that only 2 out of the 8 genes that have been cloned, rust resistant genes that have been cloned out of barley at the moment, are NLRs, right? And if you contrast that with wheat, 15 out of 20 are NLRs. So we're not sure whether this is just pure chance or whether it's an indication that in barley, maybe the architecture of rust resistance is different to what we see in other plants. And in many other plants, disease resistance, the NLRs are very common as disease resistance genes. But certainly there's only 2 out of the 8 we've cloned so far that are NLRs. The most recent of these is RPH7 that I've got a red asterisk next to, and I'm going to tell you a little bit about that now. So we've only just confirmed this in the last 2 months, but we've isolated this gene, and it's an NAC transcription factor. So that's, I think, the first example of a resistance gene to a pathogen that's fallen into that large group of genes. So the RPH7 story goes back to work done in the US in the early 70s. It was mapped to chromosome 3HS. What we found in looking at this is that virulence globally is quite uncommon. Now you might say, well, is this a durable gene? We don't really know. I guess it hasn't really been deployed in barley varieties to any great extent. There were two varieties in Australia that were released with RPH7, but they weren't grown to any great extent in the acreage. They were never popular varieties. One of the things that I found in working with the isolates and looking at these all-important phenotypes is that there was variation in the low infection type that I was seeing in barley to this pathogen. So just basically, as a generalization, I was able to pick 4 different low infection types associated with RPH7. Not just in the differential, Sabata kappa, but I saw exactly the same thing in a near isogenic stock, Bowman RPH7. So this suggested to me one of 2 things. Either there's variability in the pathogen in terms of its avirulence on RPH7. We sometimes see that these avirulent genes are partially dominant, and you get intermediate low infection types when you get heterozygosity for avirulence, or there's more than one gene at the RPH7 locus, and we're lighting it up with these different cultures. So they were the 2 hypotheses we had. And we've cloned RPH7, and we were able to actually answer this question. I'll show you that in a minute. So what we did was, this was work led by Peter Drukartis in my group, we went back to initial mapping work that was done by Beat Kaller and Kathy Foyer in Zurich, where they'd identified 4 probable genes that they thought were candidates for RPH7. And these are the ones that are up the top there from the left one. I don't know if you can see that, but the red interval there, starting with this HVGAD1 gene, and there are 3 genes to the right of that were all identified as probable candidates for RPH7. What they did was they'd done Sanger sequencing of back contigs, but they had some gaps in their sequencing. So we went back and we resequenced those contigs with those back contigs with PacBio. We were able to put together a nice contig across the entire area and then do gene prediction. And we found a few extra genes in there that they hadn't seen. We were able to, we were also able to, through recombinational genetics, remove the candidates from the one on the right-hand side of the red interval across. There's 3 genes. We were able to rule them out based on recombination. So we knew the genes sat in that red region, and we were able to predict some additional genes in there following the PacBio sequencing of those contigs. We then looked at RNA-seq, and that showed us that some of the genes that we found were actually were actually expressed. And then we did an RNA-seq expression study on those genes, and we found 3 of them were upregulated in the resistant line when those lines were challenged with an avirulent pathotype. So this was the bottom graphs were taken at day 6 post-inoculation, and these 3 genes were significantly upregulated in Sabata kappa, which carries RPA7, Bowman RPA7, but not in Bowman. So these were all prime candidates for RPA7. The next thing we did was we actually went in and sequenced those 3 genes from some knockout mutants we generated. Okay, so we generated a mutant population. We found, I think it was 10 mutants, and in 4 of those mutants, we were able to show changes in only 1 gene, SNPs and only non-synonymous changes in only 1 gene, and so that told us that this was RPA7 pretty confidently. So we then transformed that gene into Golden Promise, and we did the Rust testing, and we were able to confirm that. And not only were we able to confirm that the gene that we have is RPA7, but we were also able to explain this variation in low infection type. So here is Bowman plus RPA7 non-transgenic. This is just the near-isogenic stock with 2 different pathotypes, showing you the difference in the low infection type that I said before could be due to the presence of more than 1 gene or difference in the heterozygosity of the pathogen, right? So you can see that both still resistant, but very different low infection types. Well, it turns out the transgenic did exactly the same thing, right? So the next 2 leaves are actually Golden Promise plus RPA7, and you can see that same difference in the low infection type. So that tells us it's something in the pathogen, there's only 1 gene, and it's probably due to heterozygosity in the pathogen. Okay, and the next 2 are just the Golden Promise and Bowman controls with pathotype 5477P plus, showing the susceptible interaction. Okay, so we've been able to clone RPA7. The in silico protein modelling has suggested that it's a NAC transcription factor, and the transgenics has told us that this difference in low infection type is not due to multiple genes, it's just 1 gene, and there's something going on with the pathogen. Another thing that happened when we were doing that work that turned out to be purely fortuitous and really lucky was that I received a sample of leaf rusted barley from Tasmania in December 2020, and we, as we always do, we put this on a set of indicator lines, and I noticed a mixed infection type on Sabata kappa, the RPA7 differential, but this time it was a difference in avirulence and virulence. So I'm hoping you can see that there. The blue arrows are pointing to incompatible interactions. So this is an isolate that is avirulent on RPA7, and the red arrows are pointing to virulent pustules. So there's 2 pathotypes here, at least, one of which is avirulent on RPA7 and one of which is virulent on RPA7. And this is the first time I detected virulence on RPA7 in Australia in over 30 years. I subcultured those high pustules, and sure enough, it's an isolate that has virulence on that gene. We used that on the transgenics, and they were all susceptible. So again, that's further proof that the gene we've got and we put into the transgenics is RPA7. So the rust and the phenotype doesn't lie. What we did find, and this is why the Swiss group had difficulty in pulling this gene out, is that there's a lot of structural variation around this region. So this just shows the interval between the 2 flanking markers across, I think it's 20 barley varieties. And you can see there's huge sequence divergence there in terms of gene complement and also size. So they were working off the Morex reference, I think. I think so anyway. And you can see that the Morex is down in group 3, where there's just no homologous sequence to RPA7 or Sabata kappa. So these structural variants really stress the importance of having more than 1 genome, being able to work off pan-genomes to try and home in on these genes. We found a similar situation when we isolated RPH3. Morex, I think, had a 90 kb region that was missing in the variety carrying RPH3. So using the reference really wasn't helping us get to that gene. And it was only when, very fortunately, we realised Martin Masher had sequence data for a variety called Bark that carries RPH3 that we were able to identify candidates and then isolate that gene. So these structural variants certainly have not made isolating these genes easy. All right, who's a Doctor Who fan? Remember the Daleks? they had an expression that said resistance is useless. Well, not in resistance breeding. So we had an economic analysis done on the value of genetic resistance to various diseases in cereal crops in Australia, done in 2009. And they estimated in wheat alone more than this $1 billion a year, the value to the industry of genetic resistance to rust in wheat alone. The people that did that analysis, it was quite comprehensive and they're very respected. So, I mean, even if they're out by a factor of 50%, it's still a very strong endorsement, I think, of the approach of resistance breeding and the importance of these resistance genes in controlling these pathogens. So pulling all that together, just into a few things to conclude with. This document we put out, we try to put out at least every 2 or 3 years. It basically lists all current Australian, in this case, wheat, durum wheat and triticale varieties, with the resistance genes they carry to each of the 3 rusts, both seedling and adult plant, and then a consensus field rating, whether they are moderately susceptible, resistant to moderately resistant, or whatever. Moving across that table to this side, you'll see that there are actually 2 ratings for stripe rust. And the reason that that's there is because we have different pathotypes of stripe rust in Eastern Australia to the ones that occur in Western Australia. And so the varietal responses can be very, very different. And that information comes from our race surveys telling us that the pathotypes are different. Okay, so and that's really important. So we know there are varieties that are perform really badly in Eastern Australia because we've got virulence, but put them into WA and they do very well because there's no virulence in the pathogen there. So that document is really heavily used by our industry. I mean, apart from the fact that it's useful in making decisions about varietal selection and then management of varieties, if somebody's growing a variety that is rated as being resistant and they're seeing stripe rust, straight away they're worried and they notify us and then we can try and work out what's going on. Has there been a change in the pathogen? If there is, what varieties are going to be affected? Quite often these varieties share resistance genes, so then we can make predictions about which other ones are going to be affected. So as I said, that document really is a summation of all the genetics we're doing, all the rust stuff, rust rate surveys we're doing, the genetics we're doing. I mean, the APR genes there, the font's very small, but the APR genes there are postulated based on markers that have been developed through the collaboration, particularly by the people at CSIRO, who developed some wonderful markers for these APR genes. So I like to say that this is sort of a document that brings all of that work together and then provides it in a form that is really, really heavily used and referred to by the industry. So just then to make a couple of a few concluding comments. We know that our preemptive breeding approach in Australia has been very successful. That's been vindicated by economic analyses that have shown big savings to the wheat industry from resistance. What we're seeing is increased movement of rusts, an intercontinental movement and more of these exotic rusts coming in, and that's creating problems in the industry, and it's also creating problems from the point of view of extension and getting people to keep the faith. You know, they see varieties fall over and they say this genetics is no good. You know, we'll just use fungicides. So that then makes it, you know, more challenging to convince people that really genetics has to be the foundation of what we're doing. I'm going back to Australia next week. I'll be home on Thursday. Next Monday, I'm speaking to farmers out in the wheat belt. I've been asked to go out there to a place called Griffith and speak to farmers because they've got lots of stripe rust and they're looking at spraying crops 2 and 3 times and they're really, really not happy. So, you know, it makes our job more difficult in convincing people of the importance of genetics, apart from the obvious impacts. What we do know, and I think everyone is in agreement on this, the more genes you've got in there for resistance, the better. It's more difficult for pathogens to overcome them. We know in Australia that mutation is a major source of variability in terms of the number of pathotypes that we get, but when we get these incursions, they are very, very different. The gene cloning, it's really quite amazing. We're now developing perfect markers for these genes. So in the past, we would, you know, that table I showed you, all those varieties, we do that with pathotype testing. So we'll take the varieties and test them with 15 different pathotypes and we can postulate the resistance genes that are present in things. Now we're getting markers that'll do that job for us. And that's making that process much easier, particularly for the adult plant resistance genes that are only expressed in adult plants in the field. And they're very difficult to know whether they're present or not. That gene cloning is also opening up the possibility of cis and transgenic approaches and, of course, gene editing, looking at doing sequence variation or modifying sequences of resistance genes to try and change specificity. So that's all really exciting work. And they've actually been able to show, again, my colleagues in CSIRO have actually been able to develop technology where they can put a number of these genes into a single cassette and transform them into wheat. And so you get 4 or 5 genes being inherited as a single Mendelian trait. It works, they've got it working. But nobody's approved transgenic wheat, so it can't be, there's no pathway to market at the moment. But they've developed the technology and it really is quite amazing. On the pathogen side, we're now doing a lot of genome sequencing. Again, the technologies are there, the protocols are there, where we can actually do fully haplophased assemblies, chromosome scale assemblies for Rust pathogens. That's allowing us to look at their evolution more carefully. Hopefully it'll help us to work out what's going on with that barley grass stripe rust pathogen, for example. But it's also giving us markers where we can monitor the global spread, if you like, of these things. So we now know those 4 isolates of stripe rust, we know where they've come from. It gives us a bit more insight into entry pathways, and maybe there can be some additional regulations put in place to try and minimise the chance of that happening in future. So we're now starting to look at the spread of these things around the world. But the point I'll finish with is, we can do all this gene cloning, we can do all this fancy stuff now, but we really have to get this into farmers' paddocks. We need to keep translating the results of our research into farmers' paddocks. I'm speaking to the converted here because you folks do that really, really well, I know. But it's a point that I often finish my seminars with. It's really important that we do that and we get out and speak to the growers. So I'll leave it there. I think I'm maybe going over time a little bit, but just some acknowledgements, and if there's any questions, I'd be really happy to take them. Thank you.
So we've got a question from Nisha Bayer. Is he here?
You've got to come back here if you answer.
So Nisha asks, what is there to stop stripe rust pathogens spreading between East and West Australia? If intercontinental spread is possible, presumably spread within the continent is easier.
It is. Whenever we see something in Western Australia, you give it a year, it's in Eastern Australia. Give it another year, it's in New Zealand. And we've seen that so many times, this west to east movement on prevailing winds, going back the other way. Exactly, we don't see that so often. So we had wheat stripe rust in Eastern Australia in 1979. It turned up in Western Australia in 2002. So we were dealing with that as a big problem in Eastern Australia for a long, long time. We managed to keep it out of Western Australia until 2002. When it turned up in WA, we thought someone's taken it over there. Did the race analysis? No, they didn't. It was completely different. So we knew it had come in to Western Australia, probably from North America. But again, gave it a year, and that thing was in Eastern Australia. We predicted that. So yes, there is restricted movement from east to west, and that has important implications for gene deployment. We know some genes that still remain effective in the West.
And a second question from Ian Toth. Excellent and clear talk, Robert. Can you say something about the threat by these pathogens to UK, Europe, and how changes in trade and climate might affect this in the future?
Yeah, well, I think what we're seeing in Australia is reflecting what you're seeing here. I mean, as I said before, this with the 2 stripe rust groups, they are the dominant pathotypes here in Europe, and they are in Australia. So I'm not sure about things. I mean, it's an interesting question, is it? I stand here and I complain about all these incursions into Australia, but we're exporting them all to New Zealand and possibly other places. I don't know of any other examples of that, but it's quite possible they could be brought back to Europe. And in fact, you can have a variant within a pathotype that you wouldn't necessarily identify as an exotic incursion, but it could be just a different genotype of a specific pathotype, and that could be important. Yeah, so we just need to keep an eye on that. The question was about climate, yeah, also. So there's a concern around changes in air currents, for sure, with climate change and how that's going to impact on transboundary pathogens. There's also concerns around, I'm trying to think of it now, I know the FAO has looked at this, but there are concerns around what a changing climate might do to migration patterns in these pathogens. And certainly in Europe, for example, and I don't know if this is necessarily related to migration, but there's been a reappearance of stem rust of wheat in Europe that's believed to be associated with a changing climate. And that pathogen's just not been seen in Europe for a long, long time. So changes in climate, yeah, they will. You know, we're seeing evidence of that now and the impact on these diseases.
I think we're becoming a bit complacent to rust resistance against the cereals in Europe.
Yeah.
I had a question just in relation to what's been asked. Is there resistance to these pathogens in European germplasm?
Yeah. I mean, it's certainly with stripe rust, I would say that there's been very good resistance for many years. We've actually done genetic analyses on that in the winter wheats. Stem rust, not so much, and there's concern around vulnerability to stem rust. I think if you go back 20 years, I was here in 1995 in Germany, and I looked at the resistance of a whole selection of European wheats, and 1B1R was pretty common in those days. So there was good stem rust resistance coming from 1B1R. That's not there so much anymore. And I think there's VPM is present in quite a few wheats now. That is effective here. It's not effective in Australia. So I wouldn't be putting too much faith in the SR38, the stem rust component of VPM. SR24 is in a few winter wheats that the German folks are telling me. And what was the other one? There were 3 genes, but it's fairly narrow, the genetic basis, and there is a lot of vulnerability.
Eleanor?
There are no more questions online.
Alison?
Yeah, on the DMI insensitivity, I was just wondering how common are the DMI insensitive pathotypes in the Hordeii population? And if DMIs are the only mode of action available, and you're seeing increasing spraying because of the lack of resistance, are you driving towards a situation where you have no means of control?
Yeah, so we do, to start off with, we do have other chemistries. So there's at least one succinate dehydrogenase inhibitor, one SDHI, that's been registered, and there's a number of strobilurins, and there's a number of mixtures. So there are alternatives there that will control this. In terms of the population, it's actually present. It's an interesting story because when we found that insensitive isolate, I know the pathotype, right? And we found it in an isolate of that pathotype that was collected 2 years ago. I went back to the first collection of that pathotype that I found in 2001. So I found it originally in 2001. I think it was an exotic incursion, and it is insensitive. So we had it in the collection. We did a trace back and showed the original isolate is also insensitive. So it brought the insensitivity into Australia from somewhere else. And any isolate within that, any pathotype within that group carries the insensitivity. They are present over all of Australia now. And just one additional piece of information on that, that pathogen is the only rust pathogen we have in Australia that undergoes sexual recombination in a very small part of South Australia on the York Peninsula. The alternate host is there. And we've now got isolates that we believe have come through that thing undergoing sexual recombination. So we're now getting recombination of the insensitivity with virulence, with other virulences, but we also have some evidence of even higher levels of insensitivity coming off the alternate host. So, but there are other chemicals that they can use to control it. Yeah.
David?
Yeah, thanks. It's really interesting to see. I work on late blight, and we have sort of similar, but in many ways different challenges. One thing I was wondering about, you showed that table with the very small fonts in wheat with 3 different rusts. No,
Yep.
It's fine. I mean, it showed the complexity, and that was the point.
Yeah.
My question was, how do you translate that for growers, or who translates that for growers? Because, I mean, depending on where you are and depending on what your markets are, it's obviously quite a complex decision to make a decision on what am I going to grow this year, and can you shape that? Are you doing that strategically to allow new sources of resistance to be extended to last as long as possible? Who makes those decisions? Is it advisors, or how does that work?
Yeah, it's a huge document. You can download it from our website and have a closer look at it. But yes, I think the font size was such that you probably couldn't see a lot of the detail. There's actually columns in there that with the genes, any genes that are effective are bolded. So there are genes there we've listed for which we have virulence and nothing. So they're not. So the bolded genes are the ones that are effective, and they're contributing to the resistance rating we're seeing. And then there's another column that tells you whether the resistance is due to adult plant resistance or the seedling resistance. And the reason for that's just helps, and it's more the agronomists that are interpreting this than farmers themselves, right? Because most of our farmers have advisors. But they're looking at that and they're saying, okay, here's a variety that's rated resistant and it's got seedling resistance, so it should be resistant at all growth stages, but it's got some rust in it. So they'll phone me up and ask me what's going on. Or it's a variety that's got adult plant resistance and they're seeing some rust in it. And so they'll call me up and I'll say, so it's like often they're consulting us, but they'll say, you know, this, and I'll say, what growth stage is it? And they'll tell me, and I'll say, well, I think you'll find it should be okay because we rated it as an MR, adult plant resistance, which means it'll take a bit of stripe rust initially, but it should outgrow that and be okay. And invariably what happens in those situations is that they still spray. Yeah, because it's just, at least they sleep better.
We have a farmer in the audience who wants to ask a
yeah.
question.
Yeah.
He wouldn't spray, would you?
I'm afraid our advice is to spray as early as you can, like control. I think following on from what you're saying, AHDB produced a recommended list in the UK, and within that recommended list, there is a rust category within that. 9's the most resistant, right down to 1.
Yeah.
But it doesn't split into different variants and things like that. So it just gives that variety of rust resistant, and that's within barley, wheat, and oats as well. So the recommended list does do that, but not near as complex as what you've just shown on the screen, like.
Yeah. Well, there's actually two sources of information. So we have a similar thing with the national variety trials. It just lists the varieties and their responses. But then people, if they want to drill down into that, they can go to our document and look at that. And as I say, a lot of our agronomists actually are degree qualified, and they are very interested in this and knowing whether the resistance is this adult plant resistance, which is not really high level of resistance necessarily, or whether it's one of the major genes that's giving really high levels of resistance. And then trying to explain what's going on in their clients' crops. Yeah, but it's an enormous amount of information, but it's very well received by industry. We know people like it. And the reason we know that is because if we're late, if we haven't put one out for 2 or 3 years, people are saying, when are you going to put the next one out?
Any more questions on the chat? Malcolm?
How do you carry out your phenotyping? Is it automated at all? Image capture, machine learning?
Sorry,
Your phenotyping?
Yeah.
How do you carry it out? Is it automated?
automated?
Yeah.
Yeah, well, it's done manually, and it's a very, very high throughput system. So last year we put through the greenhouse 6 semi-trailer loads of potting mix. We do hundreds of thousands of assays, if not 1 million assays. Having said that, my funding agency has said to me, we want you to see if you can make it better by automating it. So they're funding a project to look at ways of possibly doing image capture and I suppose algorithms for interpreting responses and stuff. I think that that's very well advanced in the. Well, I shouldn't say it's well advanced in the field, but there's a lot of activity, as you would know, I think, going on in the field, and I can see that's going to be really big. To be able to capture a whole heap of data from plots at once. In the greenhouse, I'm kind of not convinced because it's such a high throughput system. We can score so much material so quickly. But having said that, at Aberystwyth the other day, they were showing me their phenotyping thing, and they're now, they've got these gantries and that, but the digital imaging is getting so cheap these days, they're going to have just hundreds of cameras, I think, just taking pictures all the time. So, I looked at that and I thought, well, I can see maybe, this is what's going to happen. So, it'd be interesting to know that our funding agency has said to us, we want you to look at this. We're going to give some funding to you, but you're going to have to bring people in because we don't have any of those skills. It's going to be really interesting to see what those people say when they look at our system.
Times are changing. So listen, I think we should probably bring this to a close now. The one thing that your presentation really reminded me of was, I know rust's a really serious pathogen, but it looks like a beautiful pathogen to work with.
It is.
Because
all different colours.
things are so clear. I mean, at least the presentation was so clear. It also reminded me of how good that rust network is in Australia, the collaborations you have with CSIRO and other, bring people together to actually do things which are really relevant to agriculture. Robert, thank you so much for coming. Thank you very much for the presentation. I had one trivial question for you before we finish completely, and that is cob the origin of cobbity?
No, I don't believe so. From Nathan.
Yeah,
Cobb.
Nathan Cobb.
I don't think so.
So Cobbity wasn't named after Cobb.
No, I don't know where. I think there's probably a place in England called Cobbity.
No, is there? Okay.
Yeah,
You don't want to be
find it's
associated with
probably
that.
at least 50% of the place names in Australia you'll find here. So we just copied. So I don't know if it's one of those. But no, I don't believe it's got a connection with Nathan Cobb. But we still use the Cobb scale. So his name lives on
Absolutely,
for sure.
absolutely. So if we could just thank Robert one more time.
Oh, thank you.
Thank you for coming.
Thank you also for being such wonderful hosts. I've really, really enjoyed the visit.
Good. It's not over. Well done, that was great. That was great.
Yeah.
Thank you very much, Robert.
This is George Laurie. George is the chair of our International Barley Hub.
Oh,
Farmer,
that is so.
not a scientist.
That's great. That's great.
But heavily involved in a company called Scottish Economy. We do a lot of research. We've got one of our field research. We do a lot of